This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 666, recorded on September 23rd, 2020. I'm Vincent Dracaniello, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's a sunny day. It's 68 degrees Fahrenheit, Celsius.
That's 20 C, right?
Yeah, let's call it that. Good enough.
And from Austin, Texas, Rich Condit.
Hi, Vincent.
Morning.
Hi, Kathy. Hi, everybody. We've got 72 degrees and overcast. We're about out of the rain. It's going to be 80 this afternoon. We're dealing with the sort of edge of beta here, but that's not done anything other than to bring us some welcome rain.
We're supposed to be 81 this afternoon.
Hey.
We might beat you.
Wow. From Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. Sounds like I'm in exactly the same weather boat as the others. It's 70 Fahrenheit here and sunny. So that's pretty good. I don't know what it's supposed to get to this afternoon.
It's like 26-ish.
Oh, okay.
It's not bad. Our guest today is a Higgins professor of radiation biophysics. And I say Higgins professor because I'm also a Higgins professor. So now we got two Higgins professors on the same podcast. And also the director for the Center for Radiological Research at Columbia University Irving Medical Center, which of course is where I am. David Brenner, welcome to TWiV.
Good to be here.
You got episode 666. I don't know if that's a significant number, but it's a cool number. We are going to talk about your work and your career. Before we do that, if you like what we do here on TWiV, consider supporting us, microbe.tv/contribute. If you want to chat with a virologist, you can go to asv.org, and there's a tab along the bottom of the homepage where you can find the link.
The top image on the homepage now. It's within the image. It's slightly different from where you might think. It's also in the rotating images, but it's in the top image now on the right-hand side. We tried to make it really prominent.
Okay. And if you really like to learn virology, take a course, virology.blog/course. David, how long have you been at Columbia?
Too long, many decades, more than 30 years.
I arrived in 1982. Is that before you or after?
That's pretty close to me. We're contemporaries.
Wow. Same age, I guess. So just trace your history a little bit for us. You must be from the UK, right?
I am indeed from the UK. Liverpool was where I grew up, just after the Beatles. And undergraduate in Oxford, then graduate work in London. And then I took the pretty standard route at the time in the late 70s to come as a postdoc to the USA, and I came to Los Alamos lab in New Mexico when they were doing cancer research, which is sort of my day job, really. I was there for 3 years, and I've been at Columbia ever since.
So your research has been on cancer most of your career?
Well, it's mainly been on ionizing radiation, that's X-rays and gamma rays, okay. Which have, as you know, a two-edged sword. They can produce cancer, but they're also being used a lot to treat cancer. So I'm in the Center for Radiological Research here at Columbia, which is an old and venerable institution. It was actually founded by a student of Marie Curie just over 100 years ago. Faïla went to Paris, did his PhD with Curie, and came back and started our center. With the at that point, it was entirely looking at the potential for radium to cure cancer. And we've been working on that problem ever since, I guess, with some successes.
So you were director of the center, but you also have a lab and you do research at Columbia, right?
Yeah.
So we of course, want to talk about UV light and viruses. You wouldn't say you're a virologist, but you've gotten interested in them, right?
No, I'm definitely not a virologist, so you'll correct me if I misspeak. I'm a radiation guy, radiation biophysicist, essentially is what I am.
Okay.
But I have a team now of people who know more than me about viruses.
Okay. So we picked two papers out that we can focus our discussion on. One of them is far-UVC light, a new tool to control the spread of airborne mediated microbial diseases, and another one, far-UVC light, 222 nanometers, efficiently and safely inactivates airborne human coronaviruses. So let's start just by having you explain what is UV light in general and what is UVC light?
And I'll come to far-UVC light. So there's nothing very special about UV light. It's a type of light that's very similar to visible light, and the only difference is the wavelength is a little bit different, and so it's invisible to the human eye. Mm-hmm. So UV light has actually been known for more than 100 years now, since the 1880s, to be very good at killing pathogens, bacteria and viruses, both. So it's been used over the years a lot. In our own medical center, it's used to decontaminate surgical operating rooms overnight, for example. And so when all the surgery is finished at 10 in the evening, everybody leaves the room and these lights are turned on, and come 5 in the morning, when people come back, they turn the lights off and you have a very clean room. The air is clean, the surfaces are clean, which is great. And so it's certainly true that UVC light does kill bacteria and viruses. The problem with that is that scenario, there were no people in the room, and that's how it has to be, because conventional germicidal UV light is actually a health hazard. If you shine it directly onto people, it's going to potentially cause health issues, skin issues, and eye issues. So it's all very well having a nice clean room at 5 o'clock in the morning, but when people come into that room, they're going to breathe their pathogens, their bacteria and their viruses into the air, and then you no longer have a clean room. So, and that translates into trying to clean any office or anything like that. You can certainly clean it overnight. You can do it with UV light. You can do it with Clorox. You can do it in many ways. But what you really want is some continuous source of disinfecting the room as people are in the room. And that's where far-UVC light comes into the story. So it's all to do with the wavelengths. So conventional germicidal UV light, the type that's been used for many, many decades, that's about 250 nanometers in wavelength. Far-UVC light, which is what we came up with the idea of using, is about 220 nanometer wavelength. So you might think, not very different, 220, 250. But actually, it's the world of difference. So in terms of killing pathogens, viruses, bacteria, including SARS-CoV-2, they're pretty similar. But the difference is in the safety. Mm-hmm. So conventional germicidal UV light can penetrate into our skin and can damage the skin and can damage our eyes also. 222 far-UVC nanometer light can't do that. It has much less ability to penetrate through any biological materials. So, for example, as you know, on the surface of our skin, we have a layer of dead cells, the so-called stratum corneum. And the far-UVC light simply can't penetrate through that. So it can't reach the living cells in the skin, in the epidermis underneath that. So that's the sense in which it's potentially safe, that because it simply doesn't reach the living cells. And with the eye, right on the very, very surface of our eye is a tear layer, which is on top of the cornea. And again, that blocks the far-UVC light, so it can't reach the living cells in the cornea and deeper parts of the eye. So that's what makes it potentially safe for direct human exposure. But on the other hand, bacteria and viruses, they're very, very small, and far-UVC light can certainly penetrate into them and kill them. So the general idea is that it has the same killing, sterilizing properties as conventional germicidal UV light, but far-UVC light doesn't have the safety issues. So it can be used directly in exposing a room where people are actually present.
So UVC is the light in the operating theater that you talked about, right?
That's correct. So perhaps we should go back and talk about the different types of UV light. I mean, I think most of your listeners will know there's UVA and there's UVB and UVC, which are simply different wavelengths again. They're all emitted by the sun. UVA and UVB actually reach us down here on planet Earth. And the reason how we get suntans, and that's why we put on sun cream to block particularly UVA. UVC, which is the one which is the wavelengths that are very good at killing pathogens, on the other hand, bacteria and viruses, that doesn't reach us down here on planet Earth because it gets absorbed in the atmosphere. It gets absorbed actually in the ozone layer in the atmosphere. So if you want to decontaminate with UVC, you have to use UVC lamps. Got it. And that's what is done in those surgical operating rooms that I was talking about. And that's what's done with far-UVC. It's just a different type of lamp. Okay.
Now, you said it was invisible, but I know that in our tissue culture hoods, the UVC lamps kind of have a blue glow to them. So what is that, just so that you know it's on? Is that an artificial color?
That's essentially just so as you know it's on. That blue is not UV light. That's visible light by definition.
Okay, it's kind of like the smell of natural gas, right? They put a smell in it.
It's the same thing. The manufacturers could perfectly well block that out. I see. But they don't because it's reassuring to know it's on. Okay. And actually the same applies to the far-UVC light that we're talking about today. Again, the manufacturers, if they wanted to, could make that completely invisible. They generally don't. They tend to include a little bit of visible light just so that you know that it's on.
Got it. Now, when I walk into a room where the UVC light has been on overnight, I can smell some ionization. Is that correct?
You're probably smelling a little bit of ozone.
Okay.
This is what you're smelling. And indeed, with these far-UVC lights, it's been extensively tested to see if there's any ozone released, and there is a minute amount, but way below the EPA's regulatory levels. So nothing to be concerned about.
So why don't surgical suites have far-UVC lights and just leave them on all the time? Would that help? Or
I think it would help. Well, far-UVC is a relatively new idea. Okay. So UVC, we know, has been around for a century. Far-UVC, we really dreamed up this idea about 7 or 8 years ago, with the idea that it has a potential to kill pathogens, but safely. So it's basically a new technology. And until the start of COVID, you really couldn't access these lamps. And if you went out to buy a far-UVC lamp, you couldn't, because there was no particular reason for manufacturers to make them. So they really went around, and so we've been doing research on it for 6, 7, 8 years now. But only since the start of COVID have the manufacturers jumped up and said, we can manufacture these things and we can manufacture in large numbers. So now there are 3, 4, and potentially more than that manufacturers that are building these lamps.
Just as a practical question, for example, in the circumstance of sterilizing an operating room with UVC, it seems to me shadows would be a problem. You can't actually get into all the corners. How do you deal with that?
Yeah, it is a problem, the shadowing problem. I mean, but for one thing, you typically don't have a single UV light. And the UV light design in a room is done in exactly the same way as the visible light. And these days, if someone is designing an office, they have professional light designers, and they have computer codes which tell them how many lights you should have and where to put those lights. So the same story with UV. You can use those same computer codes, but in fact, in the context of COVID-19, I think what we now know is that it's by far dominated by airborne transmission. So really, what we're trying to do in the COVID-19 far-UVC world is decontaminate the air. There'll be some advantages that you will decontaminate surfaces to some extent, but as you say, Richard, whenever there's something in the way of the line of sight of the lamp, that will block the UV light.
I'm also interested in how you experimentally assess safety. Are there animal models you can use, or how do you do that?
Yeah, well, so I think you've hit on the key thing. I mean, I think there's little doubt that the far-UVC light will actually decontaminate airborne regions. The issue is safety. So I think of it from a few different directions. I mean, one is the biophysics that I just talked about. That physically, we have a layer of surface of dead cells on the surface of our skin. We have the tear layer, which block the far-UVC light from reaching living cells. So that's the basic biophysics. But then the next way of looking at it is to know that there are actually regulations in place, and that UV light is a highly regulated modality. So there is a regulation about how much UV light at any particular wavelength, and that includes the wavelengths we're talking about today, that the public can be exposed to per hour or per day or whatever. So certainly the manufacturers are keeping within those regulatory limits.
With far UV, however, you're talking about basically, it seems to me, a new set of regulations because you're talking about having people walking around in an environment where the far UVC light is on. So it seems to me that there must be some way to experiment, granted, there's theory, but there must be some way experimentally to test the safety aspects of that.
Yeah, for sure. But again, just finishing off on the regulatory aspect, those regulations are for continuous exposure. They're not for.
I see.
But coming to it's all very well saying, well, there's a great theory here, but you got to prove that it's safe or do your best to prove that it's safe in practice. So there are lots of models you can use. We started off, the first safety model that we were using was basically a three-dimensional skin model. So you can grow three-dimensional skin basically in a test tube. And that's a standard technology that's been around in the burns world for a very long time. So we grew 3D skin tissue. And that includes a stratum corneum and all the different layers within the skin. And we exposed that to far-UVC light, and we could look for DNA damage inside the epidermis and the dermis. And we certainly did not see any. And that was in major contrast to when we did the same study with conventional germicidal UV light, where we certainly did see a lot of DNA damage. So you can do in vitro studies or ex vivo studies like that. But you certainly want to do studies on living beings. And our first studies were done in mice. So you wouldn't use a regular mouse because it's got hair, which essentially would block the far UVC light from reaching the skin. So there is a type of mouse called a hairless mouse, and those we use extensively. And so, for example, right now in the medical center where we both work, there are about 100 hairless mice that have been exposed to much higher doses of far UVC light than we might use in real life. And they're actually just right now hitting their 1 year anniversary of being there. They're being exposed semi-continuously, 8 hours a day, 5 days a week to far UVC light, whole body, so their skin and their eyes are being exposed. And we examine the mice every few weeks. We give them eye tests. We examine their skin and we examine their general health. And these 100 mice a year in are doing just fine. They are essentially no different from the mice in the adjacent cages, which have no UV light. Well, I say they're doing just fine. The boys have got all sorts of bites on their ears because they fight.
That's an indication that they're fine, actually.
Yes.
How do you measure? How do you give them an eye test? Sorry,
right.
This is really digressing.
No, this is great.
Well, essentially, the same sorts of, very much the same sorts of things that are done with humans. There's a technique called a slit lamp. And so we take the mice out, we anesthetize them for an hour or so, and we do all the tests, and then we pop them back, and they carry on their lives. Okay,
so they don't have to look at a chart with letters and say 1 or 2 and so forth. Okay.
Well, I'm not very good at that, so I guess they are glad.
I think it's funny that they get the weekends off.
Those are animal models. And I should say, we are by no means the only lab in the world now that's doing these studies. There are a lot of studies in Europe and a lot of safety studies in Japan too. So both in Europe and Japan, they've been doing a lot of direct studies on living human skin, where you take a little area, a patch of human skin on the hand or on the back, and you radiate that or you expose that to far UVC light, and you look for essentially sunburn, erythema. But I should say that with all the studies that have been done so far, there's really no evidence of any safety issues so far.
Mm-hmm. Can you tell? Go ahead.
That's great. So how exactly does the far UVC light inactivate pathogens? I'm sort of assuming it's something to do with nucleic acid damage, but I don't know if that's correct.
There's nothing magic about it. And it's the same we believe it's the same as with conventional germicidal UV light, that it simply damages the DNA, or in the case of SARS-CoV-2, the RNA. Nothing magic about it. And of course, what we're trying to achieve is basically that the cells, the viruses are dead in the sense that they can't divide. So all our studies on efficacy are looking at whether or not the virus can divide.
So it actually occurs to me there may be a gap in my knowledge here. This is not ionizing radiation. Is that correct?
That's absolutely correct.
So mechanistically, ionization, and this is what when I think of UV light, I think of thymine dimers. Okay, certain bases absorbing light and that making them reactive with adjacent bases, and that's screwing up the code. That's different than ionizing radiation, right?
Yeah, that's absolutely correct. The ionizing radiations, which are X-rays and gamma rays and alpha particles and beta particles and neutrons, They are much more energetic, and they can much more efficiently break DNA bonds and produce chromosome aberrations, for example. As you say, in the UV world, it's a little different, and we're talking, well, in the DNA world, we're talking about dimers being produced. But they can also prevent the pathogen from dividing, and that's what we're trying to achieve.
What sorts of viruses have you looked at, and how do you test inactivation by far-UVC?
Well, completely standard technologies for measuring inactivation. Essentially, you have let me go back. You asked what viruses have we looked at. Well, we started our studies very much focused on influenza, and we've been working on influenza for a couple of years. And at the time nobody was particularly interested in coronaviruses. And then so we basically our studies were with H1N1 influenza virus. And essentially, the experiment that we set up, the efficacy experiment that we set up, was to aerosolize these viruses, basically put them inside aerosols of different sizes, and then basically flow them over a window. And that window, we were shining far-UVC light. And then we collected the viruses, and then for each different virus, there is a different human cell that you can essentially see whether that virus infects the human cell. And if it does, then it was alive. And if it doesn't, then it's dead. And that's a completely, I'm sure you know much better than I, that's a completely standard approach to looking at whether viruses are alive or dead. So once we'd been doing that with influenza virus for more than a year, come the beginning of this year or the spring of this year, we realized that it's the same story with coronaviruses. So right at the start of the crisis, it was very hard to get hold of SARS-CoV-2. So we got hold of a couple of seasonal coronaviruses, the ones that cause coughs and colds. And that was easy for us to do because the particular laboratory where we set the system up for influenza was okay in terms of safety to do these seasonal coronaviruses. So we repeated the experiment essentially that we'd done with influenza virus for a couple of seasonal coronaviruses. Again, aerosolizing them, putting them inside aerosols of different sizes, and flowing them over in front of a window, exposing them while they're traveling across that window to far-UVC light, and then looking at what proportion of the viruses were killed at the far end. So that's essentially how the experiment was done. Pretty simple.
Mm-hmm.
So, it occurs. I'm sorry. Go ahead.
You mentioned that the tear layer on our eye will protect cells in the eye, and so I wonder if any parts of the droplet or of the aerosol can sort of block the light and protect the virus. So, do you care about the size of the aerosol or the types of aerosols or mucus or things like that?
Yeah, and it's a very good question. So, I mean, you're right that when we cough and sneeze and shout and so on, we get a huge variation in the size of the droplets and the aerosols that come out of our mouths and into the air. Now, the very big ones, they're heavy, and those droplets actually fall to the ground really quickly, within a few seconds at most, and within a few feet. And that I think the reason for our famous 6 feet rule for social distancing, to avoid those big droplets that come shooting out your mouth. So what remains are smaller aerosols and droplets, and those are the ones that we can penetrate with the far-UVC light. And obviously we do experiments where we vary the size of the droplets to see that indeed we can penetrate the smaller droplets. The very large ones, indeed we won't. But we're not really aiming our technology at that. I mean, if I stand right in front of you and sneeze in your face, you're in trouble. What we're really concerned about is a situation where you and I are in a room together. There's some coughing and sneezing, and a level of ambient aerosols, potentially with viruses in, builds up in that room. That's what we're trying to try and reduce. We're trying to have a continuous source of light, essentially in the ceiling. And we want to kill the viruses that are in the smaller aerosols that are floating around in the air, and they can stay floating around, as you probably know, for hours, literally. So the whole time we're in this room, these viruses are going to be with us.
Is there a? Go ahead, Ken.
No, it was your turn next.
Is there a time factor as well? In other words, and how do you measure it? Is the inactivation like instantaneous? Does the flow rate past the window make a difference?
Well, it absolutely depends on the dose that viruses get, and it's all probabilistic. The bigger the far-UVC dose, the larger the probability that they'll be inactivated. But again, it's not so much that we're interested in a particular virus. How long will it take to inactivate that? We're interested in the fact that in the room in our office, there is an ambient level of virus that's floating around in the air, and we just want to keep that down as low as we possibly can. So, we have numbers about, in 10 minutes at this level of UV light, you would kill 99.9% of viruses and so on. But really, it's the issue is just exposing the air continuously and keeping the level of live virus in the air as low as possible over a long period of time.
We'll get a little bit more to the results in a minute, but I'm really fascinated by the UV irradiation chamber that you have in the influenza paper. There's a nice photograph of it, and it's labeled and everything. And I don't know, Vincent, if you'll be able to if you could just put that photograph in, or
sure.
I don't know if this paper's under a paywall, or maybe David could give you another photo. But anyway, so I just want to kind of go through. So there's the place where you put the virus in, and there's the nebulizer, and then there's a thing where it adjusts the temperature and the relative humidity, and then you have the UV exposure area and a particle sizer. I think the engineers that listen to this would just be fascinated, because I'm not an engineer and I'm fascinated. But it gets to the end after the particles have gone through, and it gets to this biosampler. And I just want to have you explain a little bit about how that works, because I can't quite tell from the picture and I'm not familiar with it.
Well, basically, you've got to be able to collect the virus once it's gone past the UV light, and then we can then we want to infect the human cells, as you talked about, and you can do that in various ways. And actually, we do it slightly differently from what was in those experiments now, and we do it in a pretty simple way. We basically have a gelatin filter, and that catches the virus, and then we wash the virus off the filter. So it's really a very simple approach.
Okay. And so you can do that in some kind of quantitative way. So yeah, cool.
Yeah, that's the same approach that's used when you want to measure the amount of pathogens in a room. You have basically a pump, and you pump the air across a filter, and then you wash whatever's on that filter into a test tube.
The particle sizer on this thing, is that just measure the size of the particles, and you can generate different sizes? Or does it actually, or can you sort them?
No, we can't sort them, and it's basically measuring the distribution of size. Okay. But we can use different aerosolizers to produce different distributions of aerosol sizes.
Okay.
It seems to me that you have this temperature and relative humidity meter it seems like you could actually also look at the effect of those parameters on infectivity, right? Temperature, relative humidity, because that's thought to be a component of seasonality of viruses.
Yeah, well, we do. Basically, that affects the distribution of aerosol sizes. At least from our perspective, that's what temperature and humidity do.
Yeah.
So yes, I mean, but we try and in our experiments at the moment, we're basically trying to keep those under control.
Sure. So it. I mean, it seems to me that you'd have to do some kind of an experiment to test this, a trial of some sort. And people always say, well, we can't do random clinical trials of face masks, right? But can you do that with this? Can you have 2 rooms that are out in public with people walking through? And I just don't know how you'd set it up. What do you have to do to show that this works in real life?
Well, I'd say we are working on this, and I would also say it's not easy because the things, I mean, speaking purely as a scientist, what I would like to do is to have a room. I would release some level of some amount of SARS-CoV-2 virus into the air, and then I would do measurements with and without the lamp. But of course, you obviously can't do that. So you have to. So the first thing is you have to accept is you're going to have to use some surrogate for SARS-CoV-2. Mm-hmm. And I mean, there are viral surrogates that people use kind of within the laboratory, and phages essentially. But even that, you can't realistically release some phages into a coffee bar and try and do a study with and without the lights. So it is actually very hard to think, well, how can you actually test its efficacy? Because and the real issue is that you can't release any known amount of pathogen into the air. So you have to use the natural pathogens in the air. So the air in your office and in my apartment here have a certain level of pathogens in them. And naturally speaking, there'll be some influenza virus, and there'll be some, all sorts of viruses that will be around at low levels in the air. Hopefully no SARS-CoV-2, but maybe some of those too. So the idea is we won't release anything into the air artificially. We'll just measure the level of whatever viruses are in the air with and without the UV lamps present. It's not an ideal solution, but there is, as far as I can see, there is no ideal scenario for actually measuring the efficacy of this approach. I mean, you can certainly estimate it indirectly. I mean, we know from the laboratory what level of UV light kills what level of SARS-CoV-2. So you can calculate what that would mean if you had a certain level of far-UVC light in a room. But to actually validate that in a room situation is actually quite difficult. One approach that we were taking with influenza virus, and this was before the SARS-CoV-2 disaster hit us, was to do some animal experiments. Essentially, imagine you have two cages, and let's imagine they have guinea pigs in them, and you infect. So there is airflow between these two cages, which and you infect the guinea pigs in one of the cages with influenza, shall we say. And then you look at the animals in the other cage, which can't physically touch the animals in the first cage, and see whether they actually get influenza or not. And you can do that with and without UV lights and across either of the cages. So that study actually was just about to be done when the calamity hit. So in principle, you could do those experiments with SARS-CoV-2. You can't do them with guinea pigs because I don't think guinea pigs get SARS-CoV-2. So you have to have an animal model, and a ferret is one animal model that you might be able to do these experiments in with SARS-CoV-2. Or these days, you can get humanized mice, which are which do have the right receptor in, so for SARS-CoV-2. So in principle, we might be able to do those studies with humanized mice that are sensitive to SARS-CoV-2, because mice in general don't get COVID-19.
I'm not sure that mice can transmit by air. Ferrets can, and I think also hamsters can as well.
Well, Syrian hamsters are another possibility for SARS-CoV-2. In fact, you're quite right. It's unclear whether that mouse experiment would work as I described it. So either ferrets or Syrian hamsters for SARS-CoV-2.
So I noticed that most of your examples are with coronaviruses or influenza, which are both enveloped viruses. Are there any sort of factors of the pathogen, the type of virus, structure, anything like that influence how susceptible that pathogen is to inactivation by far-UVC light?
Well, generally speaking, by far the biggest factor is the genomic size. I mean, basically how much DNA or how much RNA there is in that virus, and all the coronaviruses are about the same in terms of their genomic size. So that's why we felt we were pretty comfortable using seasonal coronaviruses rather than SARS-CoV-2 for our studies, and believing that it was going to be the conclusions would be applicable to SARS-CoV-2. But I should say that since we started this, there has been a publication on far-UVC light and SARS-CoV-2 coming out of Hiroshima University in Japan. And it was very much as we expected. Its sensitivity was pretty well the same as the other coronaviruses. So, I don't think, apart from how it looks, that we really need to do any more studies with SARS-CoV-2. I think the studies that we can do much more efficiently and easily with other coronaviruses will serve us just fine.
In fact,
that kind of goes along. That goes along with what we talked about last week, where we were talking about a paper that used the seasonal coronaviruses and their long-term immunity or short-term immunity, as it were, as a surrogate for SARS-CoV-2. And that's exactly what you've done here, is use the seasonal ones. And it seems a logical assumption that they're going to behave similarly.
I mean, just in practice, it is very hard to do studies with SARS-CoV-2 because they need to be done in a BSL-3 facility, which means essentially that I can't go in a BSL-3 facility because I'm not trained and I don't have FBI approval to do that. So to be able to do work with the other coronaviruses, which don't need a level of security no more than for influenza, is a great advantage.
I just wanted to point out that you mentioned the size of the genome, and in fact, so the bigger the genome, the bigger the target it is, and it's one hit will more readily inactivate a big genome versus a smaller one, right? So,
that's right.
Okay, so I want listeners to understand that big is good in this sense as being a target.
So following on Brianne's question, how the genome is packaged, does that make a difference whether it's an enveloped or a non-enveloped virus? Or for that matter, maybe the big question is, does it work as well on bacteria as viruses?
Well, just to start with your question about how it's packaged, the answer is I don't really know, but I would very much doubt it. I think it's just the amount of DNA or RNA that's going to be relevant. Sorry, I've already forgotten your other bacteria.
Have you tested bacteria with far-UVC?
Yeah, we And this whole project actually started with bacteria. I mean, we were looking for a means to inactivate drug-resistant bacteria, and we had envisaged, and we still do envisage, this being in use during surgical operations. So you'd have a far-UVC light basically shining onto the surgical wound, so that you'd be able to kill all the bacteria that are floating around above the wound and potentially ultimately going to settle onto the wound. That's really how we started this whole thing. And we were working on that a couple of years, really, before a light bulb hit that we could actually use this for viruses too.
It was a UVC
in general,
light bulb.
Yeah.
Sorry, just to finish the point.
Sorry.
In general, bacteria are less sensitive to UV than viruses. So the amount of UV light you need to kill viruses is really very low, extraordinarily low.
Kathy, you say your thing. I think that's got to be.
Oh, it's just that when you said a light bulb hit you, as a UVC light bulb in particular.
I didn't have a UVC light bulb at the time.
So actually, your surgical suite scenario brings to mind, if you have it shining into a wound, okay, now you don't have the skin barrier anymore. Is there enough like moisture or something like that to protect the tissue?
Well, first of all, I mean, generally speaking, you don't go around with open wounds. You put a Band-Aid on them or whatever. But yes, I mean, a wound is a pretty complex thing. There's all sorts of goo and stuff and fluids floating around inside that wound, and probably they would protect the wound. But I would say that even on the level of an individual cell, human cell, the cytoplasm actually provides quite a lot of protection from the DNA inside the nucleus. So as well as protection at the tissue level, there is some protection actually within a cell.
So I guess the elephant in the room is, right, we're in the middle of a pandemic. It doesn't look like it's stopping anytime soon. Can we get these lights into places where they might make a difference, or do we still have to keep doing experiments for the next year?
No, I mean, these lights are starting to be installed in public settings. I saw a photo the other day of the elevators in the Seattle Space Needle, and all the elevators have these far-UVC lights inside them. I must say that's a very good application, because I don't know about you, but when I'm in the medical center, I hate going into the elevators. I feel very uncomfortable. So, I mean, that's an area where you're really very close to your fellow human beings. And there are many other settings where they're starting to be installed, and including our own medical center, in fact. I noted the other day that the Miami Dolphins, the football team, has them installed in their locker room. But the issue has been, till very recently, these lamps haven't been manufactured in large numbers, and so until SARS-CoV-2 came along, there were a couple of manufacturers that were interested, but they weren't exactly going into huge production rates. Now they are, and there's 2 or 3 companies that certainly by the end of the year will be producing numbers in the hundreds of thousands and millions of these lamps.
So are there standards already for how long or what power of light could be in a room, or is it just the Wild West in terms of people
No,
buying
by no
these
means.
lights?
UV exposure to the general public is very highly regulated.
Even this 222.
It's essentially all wavelengths. I mean, what is in the regulation is a so-called action spectrum. So it's a spectrum as a function of wavelength of what level of UV light is acceptable at that wavelength. Okay. And that certainly includes 222 or 220 nanometer light, which is what we're using. So it is regulated.
So is it possible for an individual to get these? So is this something that you can imagine people buying for their homes, or is it something I can, get and have in my bag when I go on the train? What level of availability is there for the public?
Well, I mean, I see it much more, at least at this time during the pandemic, as more relevant to public spaces, occupied public spaces, than I do to your home, because you've got other protections in your home. You know about the other people in your home. And the issue we have when we go into a public place is we don't know what the status of all those other people that are hanging around in our office or in a theater or in the gym or whatever. So I see it much more relevant to public places. In the very longer run, I can see it being used in homes. And let me just add a lighting comment. So in terms of our visible lighting, when we were growing up, we had regular bulbs with little filaments in them. These days, we are moving over to LEDs. And the same applies in the far-UVC world. So the far-UVC lamps that are being produced right now, they are basically a gas-based lamp. So it's a glass thing with, in fact, krypton chloride inside the glass. And they're moderately difficult to make and not super inexpensive. If you could replace those with LEDs, which are much easier to make, and you can make them in huge numbers, the cost would dramatically come down. And I think at that point, you can imagine them being used in homes. That's still a little ways off. LEDs started their life at pretty high wavelengths. And over the years, the wavelength that LEDs can be used for has come down and down and down. And right now you can get 250 nanometer LEDs, but you can't get 220 nanometer LEDs. And the physicists are working diligently to make those things. And I actually have some of those I have a 230 nanometer LED lamp in my lab, but it's very, very low intensity. But it does work. It does the job. So I think that's the future. And when these LEDs come along, then I think you can have these lamps in your home.
So, in fact, all the light you see on me here is LED generated. Yeah. And in the back, you can see I have a far-UVC lamp, obviously. Right? So I get the sense that this is slowly being deployed. Elevators, doctor waiting rooms, for example, where there are a lot of people.
Dentists are very interested, I might say.
How about public transportation? I would think subways and buses and stuff might be an appropriate place.
Yeah, well, the MTA is interested. Okay. So what the MTA have done, that's the New York transportation system, right now is they've set up conventional germicidal UV lights in their subways and buses overnight. So the MTA at this point in time closes from 1 a.m. to 5 a.m. So they're using conventional germicidal UV lights during that time between 1 and 5 to decontaminate their buses and trains. And that's a good idea. But again, as we discussed, come 5 o'clock in the morning when people start getting in those buses and those trains, they start getting recontaminated. So again, what you would like is something that you can have continuously on. And the MTA is interested, and they're talking about a trial with far-UVC light.
Okay.
I think for the MTA, the subway platforms would also be great, where people congregate, and you could have on the ceiling these lights and continuously irradiate. I think that would be great, not just inside the trains and buses, of course.
Yeah, I agree. If you think about it very quickly, you can think of just a huge number of applications. Theaters, for example, all the theaters are closed right now. If in principle that this might be a way of opening them, restaurants, schools, and all sorts of transportation like airplanes, for example.
Gosh, could you imagine classrooms would just have these on all the time? It might really make a difference. But what is the timeline for really massive deployment? It's not by the end of this year, is it?
Well, by the end of the year, I think the companies will probably be in the millions of lamps in terms of their production. And I keep hearing of other companies starting up, both in this company and in China, both. So I don't think I have a very good answer to your question, but we'll certainly by sometime next year we'll have very large numbers of these around, and I would say, I mean, that this is not simply something which is in the fight against of COVID-19. As I mentioned, it's part of the fight against influenza. So it would be very nice to have these ready for the next influenza season, which we may or may not, and the influenza season after that, and the influenza season after that. And also, in some sense, we'll be ready as much as we can be for the next pandemic, which comes along whenever it does, because surely sometime in our lifetime there will be another one.
Well, I mean, all sorts of airborne viruses, not just influenza and coronas, but rhinoviruses, adenoviruses, para-influenza viruses. I mean, it would really get them all.
Measles, I think about. But yeah, so it's a one size fits all, which is sort of very different from the vaccination approach, for example, where it's very specific to a particular pathogen. This is generic to all pathogens.
Wow, I really see a day when everyone's homes are full of these things, and but I do hope that we don't find some untoward effects after a few years.
Well, again, I mean, if it can't reach the living cells, it's very hard to see that you can have any effects at all. And if it can't reach the stem cells, which are down in the basal layer of our skin, which it absolutely can't, it's hard to see that you could have any long-term consequences.
Can you imagine selling points of a home, built-in far-UVC lights in every room?
I'm too busy imagining it with daycare centers.
Yeah, that's another.
Okay.
Yeah. Well, nursing homes, which as you know is one of the major issues in the whole COVID-19 crisis, is one that we think about.
Rich and I, back in February, were in a retirement home, and we passed a room where there were 50 people sitting around tables playing cards. Remember that, Rich?
Yeah, oh yeah.
And we said, this is a perfect transmission scenario, but all you'd need would be far-UVC lights on the ceiling, and it might knock it out. Of course, the cards are going to be contaminated, right?
Well, again, it's, I think it's so much dominated by surface transmission, and it's been interesting, and I'm sure you followed the story of how the accepted wisdom has moved from it being a surface-based transmission to an airborne-based transmission, and there are still people who disagree, and obviously it's both really. But the question is what's really dominating the transmission, and seems to be the evidence is building that it's airborne.
Anyone have anything else before we say goodbye?
No, this is great. The only thing I would say is that I'm just continuously fascinated by human ingenuity. Okay, and how creative people are, and kudos, David. This is wonderful.
Thank you, Richard.
Yeah, I am sold. I think this should be put in most places as quickly as possible, and I think it'd make a big difference. So, David, thank you so much. David Brenner, Columbia University. Appreciate you coming on, and good luck.
It's been my pleasure talking with you.
Bye-bye.
Thank you very
so
much.
So,
that was cool.
That was great. I just cringed every time he said living and dead, but I didn't.
That's okay.
You're good. We all smiled at one point.
Yeah, right. I noticed that.
The other thing I wanted to say is this FBI clearance. Not every BSL-3 requires FBI clearance, only if you have select agents in your BSL-3. Like SARS-1 is a select agent, and if you have that, you need FBI clearance. But not every. We have 2 BSL-3s at Columbia. One of them has select agents, the other does not. So just to let you know.
I'm also impressed with how important it is, or at least the impact it has on me to actually talk to these people. Okay?
Yeah.
Because you read the papers and you think, it sounds like interesting, but, just another idea. But you talk to them, and this is very well thought out, very well researched, makes a lot of sense. Okay. Guys talking real science. It's good stuff.
Yeah, it's nice because sometimes when you are reading about something like this, you think of caveats to it. And it's nice to be able to ask those questions and have someone actually address them.
Mm-hmm.
No, as I said, I'm convinced. I think this should be deployed big time. It sounds like it is, but it could make a difference. And the thing is, how would we know, right?
Exactly. I mean, this goes back to the parachute thing, right?
Yeah.
We can't necessarily do random controlled trials on everything. You're not going to, and even if you could, you put it on some subway cars and not other subway cars. How are you going to be able to? You're going to contact trace everybody who goes on the subway?
No,
I don't think so.
But there's always going to be a place where you can encounter viruses where there's no far-UVC bulbs. So I'm not sure if you would see an overall effect or not. We'll see. I mean,
I really like the elevator thing. That makes perfect sense.
So my wife goes to physical therapy, and she says, I'm really worried because the waiting room is too crowded. I don't want to go. And this would be a perfect solution, right? To put it in there, and that could really cut down transmission, and together with masking, right?
I like the dentist idea as well. I remember in our previous episode where we heard about all of the aerosols made during dentistry. That this could make a big difference.
Yeah, they could, the light that they shine on you, they could incorporate this into it, right?
Yeah.
Right. Yeah. A friend of mine was saying that she was at in a waiting room for a doctor's appointment, and these people had their masks, under their chins, or they just kept touching their masks all the time, and it was driving her crazy. And so, for the moment, they need to have different capacities for waiting rooms if they can. And it doesn't sound like they are. When I went to my dentist, I was the only one in the waiting room. So,
It's funny. I chose blue for my, background. It's perfect for this episode.
And I have to admit that I never realized that the UV light was invisible.
I didn't know that. Yeah. I thought the blue was it, right?
Yes.
But it makes perfect sense.
And I immediately went to the thought of, just like that gas they put in natural gas. And we all thought the same thing. So we have, as you all do, we have cell culture hoods with UV lights. And when you're not using them, you turn them on, you leave them on overnight. And they burn out, and they're these long bulbs. Pretty expensive, if I remember. So that's what he was talking about. Oh, it's so cool. Very good stuff. Yes.
Well, folks, there you go. If you're involved in decision-making, consider getting far-UVC light bulbs. I wonder if you Google them, you can find, let's just see, far-UVC light. I wonder if you can put them in a regular socket, or if they need something.
I was thinking about that. It sounds like the subway's all set up.
Yeah.
I would think that it would be a pretty standard
for the germs. I mean, they just need to design them and build them that way, especially if they're going with LEDs.
Yeah, there are. You can buy them. Here's one. For $90. It's this little handheld thing, Brianne, that you can carry with you.
I'm ready.
Brianne.
Go ahead.
It's for external use only, Brianne.
Yeah, let me show you this. I will show you.
I was going to ask you, Vincent, if it's through Amazon, then people need to use the TWiV affiliates link.
This is just one company that I found. Here it is. It's a little handheld thing for $90. UVC kills 99.9% of germs in 30 seconds. Wow. So that's one. There are quite a few others.
So click on specs. I want to make sure it's got the 220.
Specs.
Yeah.
Where is the
265 to 275?
That's
not
right.
Be good.
That's not the right wavelength, right?
No, right.
No.
UVC LED wavelength. No, that's not right. It has to be 220. Then they have UVA, which you can tan yourself with it, I guess.
Yeah.
Wow.
I don't want to tan myself, and I don't want to use this wavelength on the train.
Interesting. No, it's not the right one. So, folks, if you're going to look for these far-UVC, 220 nanometers, right? That's the wavelength?
Yes. So we need to get everyone on board with both Michael Minna and David Brenner, and then everything will be all set.
Right.
Wow. I'm impressed. I'm very impressed.
By the way, this just occurred to me, so I have to say this. I got this, Facebook memories.
Yeah.
Mm-hmm.
Yeah.
Today's Facebook memory is from 8 years ago with you and me and Alan dressed up in spacesuits at
oh my
Boston.
Gosh. That's that long ago, huh?
Yep.
Yeah,
right. And tomorrow, I think, is, the TWiV anniversary, September 24th, 2008.
Yeah, you're right. That's right, because it's also our older son's birthday, and I know every year when TWiV was first released, episode 1, September 24, 2008.
Mm-hmm.
Me and Dixon for going on about West Nile virus.
Yep.
Holy cow. All right. Now let's go. Let's do some brevia. Kathy, you have some mask fogging tips?
Yes. So I wasn't on the show Friday when you guys were going on and on about mask fogging. And, when I was out walking, I was like, wait, I've got links. I know the answers. So there's a really good YouTube video of a surgeon. He's really talking to medical students and nurses about how to put on masks and things, but he's very detailed about how to bend the wire, how to put it under your glasses, how to fit it under your chin, and how to make sure it's fitting. But then he also points out that you can use 3M micropore surgical tape. And so we bought some. I bought some for myself, and we got some for the lab. And so I would just give one caution. He uses a small piece on his mask, just under his eyes. And if you go too far, you probably don't need to do a whole lot. And this skin is really pretty sensitive, and so when you pull it off, it's going to hurt briefly, but it leaves no marks. So people in my lab who are wearing glasses, which is all of us because we're wearing safety glasses, are using this, and this helps a lot. So
that basically closes the gap at the top of the mask. Is that right?
Right. And then by doing it underneath your chin, I have this feeling that maybe, when you breathe in and out, it may have more of it going down than up. And having the glasses over the mask is important. And then there's 2 New York Times articles. So those are from April and June. So it's kind of been on my radar for a long time, and that's why I was shouting when I was talking.
I would just like to see people wear their masks properly in the first place.
Yeah.
I don't get it because it's such common sense, it seems to me.
Yeah.
Goes over your nose, dudes.
Yeah.
Mm-hmm.
Yeah.
All right. And you had also about a funding for global virus hunting article. Is that right?
Right. So this article came out while I was on my mini vacation, and I thought for sure that it would come on to the show at some point. And every time we talk about the funding for EcoHealth Alliance, I think, we still haven't talked about this article. So it's in the New York Times, an article by Donald Neal Jr. and Thomas Kaplan, and 2 different efforts to revive funding for some virus hunting. And one is called Stop Spillover, and it's going to start in October. And it's ostensibly not to replace Predict, which was the thing that was discontinued 1 year ago. And that, I don't remember the agency behind that. But the other one is NIAID. And the NIAID one is called Centers for Research in Emerging Infectious Diseases. And there are 8 centers that are receiving initial grants. EcoHealth Alliance is one of them. And in the Stop Spillover, there's 5 entities that are getting funded, and EcoHealth Alliance, according to the New York Times article, is there. And then there's also a candidate, Biden is proposing to restore PREDICT even. So
so someone is informing him that it could be a campaign issue.
Right. Well, he's getting advice from the creator and former director of PREDICT. So there's hope for these virus hunting programs to get more funding in the future.
I didn't think it was going to go away forever,
Right.
So PREDICT.
Too good of an idea, and it's also too cool.
This is a great article. During its 10-year existence, PREDICT trained 5,000 scientists in 30 African and Asian countries and built or strengthened 60 laboratories to find animal viruses. They collected over 140,000 samples, and the global director, it was Jonna Mazet, who I interviewed a couple of years ago about PREDICT, and so I'll put a link for that in the show notes. This is a great program. And this is a good article. Yeah, I'm sorry we missed it. They have a cool picture of a bat right at the top there. Tiny bat.
Well, we didn't miss it because we got it now. Thank you, Kathy.
Now. Okay. Sure. I wanted to mention an article. So just last week, well, I've been doing chats, whatever, and I always at the end, sometimes they want me to talk about communicating science, sometimes science. If I do a science seminar at the end, I always talk about communicating science. And so I did 2 in the past week. And one of the slides I picked up last year, June 2019, there was an op-ed in the New York Times entitled, "We Must Prepare for the Next Pandemic." And when I saw it this past week, I'm like, oh my gosh. And this is by Bruce Schneier. And he writes, "When the next pandemic strikes," this is June 2019, "we'll be fighting it on 2 fronts. The first is the one you immediately think about, understanding the disease, researching a cure, inoculating the population, right? The second is new and one you might not have thought about, fighting the deluge of rumors, misinformation, and flat-out lies that will appear on the internet." And I thought, oh my gosh, how prescient was this? It's happened.
Yep.
My only comment on that is that the only thing that's new about that is the internet.
Yeah,
okay.
That's it. Yeah.
Rumors, misinformation, and flat out lies have been around since, they're part of humanity.
Yeah, it's true.
Yeah.
No, the
they
internet
can just
made
travel a lot further.
They can travel further.
The internet
it is a problem.
The internet facilitated their widespread and rapid spread for sure. But yes, they were
Change
always
the R naught of the rumors.
I have to every now and then. I have to kind of sit back and just sort of chill, and not really chill, but kind of try and chill, and realize that this problem of rumor, misinformation, and flat out lies, that it's not going to go away. That's part of what we're made of. Okay.
Yeah.
And it's just a constant struggle to try and communicate the truth in the face of all that, and point out what the lies are, what the information are, help people understand that this is going to be going on all the time. So you have to learn on your own to discriminate between what's true and what's not.
And people tend to be pretty receptive to those types of things. They're sort of excited to hear from an expert and excited to get some assistance on that.
Well, that's why TWiV is done very well, because people recognize that we generally try and do that, right? And the only thing that I don't understand is why more people aren't listening. Why aren't 60 million?
I think about this kind of thing when they talk about foreign entities meddling in the election. Okay, now that meddling is by and large meddling in the form of messing around with the information that we get, and people taking measures to try and discover this and to counteract it, and et cetera. But to me, the fundamental way to counteract that is to educate the population to be able to discriminate between real information and bogus information. And that seems like it ought to be straightforward, but it's not.
No, it's really not. I've got some classes where we focus on that, and it's really not straightforward.
All right, I have a trio of articles on infections of SARS-CoV-2 associated with airplanes. So there is I always check CIDRAP, all right, and I recommend it to you, Center for Infectious Disease Research and Policy at the University of Minnesota. It's a really nice compendium of information about infectious disease. And I have to tell you a secret. So the director is Michael Osterholm, right, with whom Peter Palese and I had a conflict at a New York Academy of Sciences meeting years ago, right? And I generally think he's doing good stuff, but he has a publicist, right? And she emailed me. Well, first of all, when the ASV was at the University of Minnesota a number of years ago, I tried to get him to do a TWiV, and he basically wouldn't do it. This was after the New York Academy brouhaha. And then a publicist emailed a couple of weeks ago, and she says, "Oh, I have a guest who would be a great match for your show." And I get these all the time now. So this person is an expert, that person. And she said, "Michael Osterholm." And I said, "I'd love to have him on, but I have a feeling he's not going to want to come on." And sure enough, he didn't. Anyway, if you want to come on, Michael, we'd love to have you. He's really, I think he's doing good stuff.
That's too bad. It would be fun to have him on. I've seen him a couple of interviews on this subject, and he's very direct and very clear and very good on this topic.
We had a disagreement over avian influenza H5N1, but I think everything else he does is very good. Anyway, there's a nice article which summarizes these three publications basically on infections on airplanes. So the first is a emerging infectious diseases article, transmission of SARS-CoV-2 during a long flight. And this one is a flight. Well, they looked at a cluster of cases on a 10-hour commercial flight among passengers on this flight. And I'm trying.
Is this the one from London to Hanoi or something?
Yes.
Yeah, that's London to Hanoi, 10-hour flight.
I love these papers.
But
I love these because I love the seating charts and that kind of stuff.
And this one now was in March, where they didn't require mask wearing. Okay, so they traced 217 passengers and crew to their final destinations, interviewed, tested, and quarantined them. And amongst 16 persons who were SARS-CoV-2 positive, 12 were passengers in business class. And there was the only symptomatic person. So there was a person probably sneezing and coughing and whatever. And 12 of the 16 people who got infected on the flight were in business class. So they say seating proximity was strongly associated with increased infection risk. So you can see where everyone sat and who got infected, basically. And I guess the other 4 were back in the cattle car, right, where I sit. And I don't know, sometimes people use the bathroom in business class, even though you're not supposed to. They do anyway, and nobody says anything. And they may have caught it on their way back and forth. I don't know if it was the air, supposedly it's filtered, so it wouldn't be transmitted that way. But that's an interesting observation. So you can be infected on a flight, and especially if you don't wear masks. They don't actually say who wore masks and didn't. I'm not sure. No,
they couldn't find that out.
Yeah.
At least in one of these papers, they said they tried to figure that out, and they had no way of figuring that out.
Right. And then another emerging infectious diseases article, in-flight transmission of SARS-CoV-2. This was a flight from Boston to Hong Kong. And 4 people with SARS-CoV-2 traveled on this same flight, and they were subsequently diagnosed positive with the exact same virus, so they probably got infected on the flight, although we don't know the source of this, right? But it's a 15-hour flight, had 294 passengers and so forth.
Now, 2 of those people were husband and wife and were probably both infected when they got on the flight.
Yeah.
All right. And the other was a cabin attendant once again, I think the message might be don't fly business class. At any rate, the other, that was a joke, by the way. The other, one of the other individuals was a flight attendant in business class. And the other individual who was infected was a flight attendant elsewhere, but a friend of theirs who may have had other interactions with him, I think. At any rate, there were pretty strong associations amongst all four of these cases. And it didn't spread any further within business class on this flight.
Now, one of the things that's mentioned, I'm not sure which of these three articles, but they say, in business class, the seats are farther away and still, you got transmission. So that means you really should skip seats in the back of the plane, right? Because I'm not sure they are.
Do we know whether the couple in this case were symptomatic when they were on the plane?
I think they were not.
On March 10th, let's see, the flight was when? March 9th. March 9th. So they arrived in Hong Kong on the 10th. And on March 10th, the married couple had fever and productive cough developed. So looks like just on arrival. So, and then the third patient was asymptomatic, identified through contact tracing. So that passenger did not have any symptoms.
In the first case, the flight to Vietnam from the UK, the individual, the index case, that individual was symptomatic on the flight, and in fact was coughing and et cetera. So, they, even though they didn't have a mask rule in place, my understanding was that they were at the time screening for symptoms, at least by asking people, because one of the comments they make in that paper is that, well, that's not going to work.
Well, I've always thought that.
Yeah.
Well, I think if you're obviously coughing and sneezing, you shouldn't fly, right?
Right.
I mean,
but people do anyway. That's the point.
Yeah.
Well, it's also like those questions when you have the enhanced TSA screening, and did anybody ask you to bring something along? And, if you were nefarious, you're going to lie.
Right.
Yeah, exactly. I know. People get through. So now, of course, you have to wear a mask. And so it will be interesting to see if there are any transmission reports. This is quite a few months after it happened, so it takes time for this to appear. But I'm thinking now, boy, you should put those far-UVC lights in airplanes, right?
Yeah. Well, my take-home message from this is that these transmissions look like they are proximity related. Mm-hmm. Okay, the closer you are to an index case, the higher the probability that you're going to get infected. And I don't see, other than the fact that you're putting a lot of people in a confined space for a long period of time, I don't see anything special about airplanes. Okay, it's not like the virus is getting into the air handling system and infecting the whole airplane.
But you are there together for a really long time, longer than
that's right.
Typical, right?
These are long flights that are being examined here.
Yeah.
No, I was sort of wondering whether one message from this might be that you should take, 2 shorter flights instead of one longer flight.
Yeah, I was thinking about that because, among other things, then at least you scramble up the people that are on the airplane. You're not sitting next to the same people the whole time. Yeah.
And the last is a publication in a journal that I'm not sure I've ever looked at, Travel Medicine and Infectious Disease. It's in-flight transmission of COVID-19 on flights to Greece and epidemiological analysis. And of course, you cannot transmit a disease. You can transmit the virus, folks. Just saying. 5 cases of probable in-flight transmission. So the point of the previous 2 is that clearly you can have transmission in flight, right? Which shouldn't be surprising, but it's good to document. So here they looked at records for passengers arriving or departing from Greece from February through March. They did contact tracing of these passengers, 18 international flights, 2,200 passengers, 110 crew members with, destinations, origins all over Europe. And there were, in these flights, 21 index cases, 891 contact traced cases. 6 cases were symptomatic during the flight. Of the 891 contact traced cases, 4 passengers and 1 crew developed laboratory-confirmed infection, and they traveled on the same flight with 2 COVID-19 cases. So just another indication that you can get infected on a flight.
However, to me, that the numbers here. I mean, it's not quite as a granular study as the others.
No, it's not, right.
But the numbers were fair. I was impressed with how low the numbers were. Okay, 891 contact cases, contact traced cases.
Mm-hmm.
Well, maybe I'm misunderstanding this. Contact trace they say contact trace cases. Those aren't people who were infected, are they?
No.
Those are contact traces.
Yes.
Yeah, I think those are people who sat less than 2 meters away for at least 15 minutes, or crew members who were near them.
Right. And following those people around, do I see this correctly? They found just 5 cases.
Yeah, that's right. That's right. Laboratory confirmed.
That's laboratory confirmed. I was impressed with how small those numbers are.
Well, it's February 26th through March 9th, so it's a pretty narrow window, and it's pretty early on. I don't know offhand what the situation was at that point in Europe, right? That's where mainly these flights are. It might have been lower. I don't want to look it up now, but that might be part of it.
Well, they said they had 21 index cases.
Yeah,
that led to these.
Right.
So depends where they're coming from and what the caseload was in those countries. But we'll see more of these, I'm sure.
Have any of you guys flown? No.
Last time I flown was February. I went to Ohio. That was it.
Yeah, I flew in early March.
Yeah. So no, I'm not going.
Because I don't know what the situation with regulations is now. Maybe we need to get an airline person on the show.
Now, the last pilot who wrote in said that JetBlue required masks, but they couldn't enforce it because they don't want to have fights on the plane. But they put you on a list that you can't fly if you do it once, which doesn't help if you don't wear a mask on a flight. Just because you don't want to. That's not fair.
There's been at least one case of a flight returning to the gate.
Yep.
Oh yeah.
Yeah, I heard about that yesterday.
I don't know how much they space out. Skip a seat in between. I don't know about that.
I think some airlines do, some don't.
Yeah, Delta still does.
Anyway, if you're a pilot or attendant of some sort and you want to let us know, we can keep you anonymous. We'd be curious to know what's going on. All right. Now, here's an article which I find just amazing. All the wrong moves in all the wrong places. This is a opinion piece from Science magazine, and let me open it here. Here we go. So the authors are Michael Yaffe and Christopher Barrett. Oh my gosh, I read this and thought, this is all the stuff we've been saying. And so I immediately emailed both of them and I said, do you want to come on and talk about this? Because I think this is just great. And I want to briefly summarize it because they may not want to come on. But they say there have been 3 major systemic failures that together block progress in disease control, containment, and treatment. This is globally, not just the U.S., right? Although they are in the U.S., they're both at MIT. These failures reflect a perfect storm of governmental ineptitude, scientific ignorance / misprioritization, and the abdication of medical training to market-driven forces. I just want to ask the 3 of you, did this resonate with you at all? Or am I the only one that thinks that this is exactly what's the problem with what's going on?
Oh, yeah.
Oh, no, this is spot on.
Yeah.
Not only that, but I appreciate the fact that they spread the blame.
Oh yeah.
They identify. It's not just one force or another. As they say, it's a perfect storm.
They're wishful thinking that COVID-19 will mysteriously vanish is both unfounded and dangerous. And they say blame for the response also resides with scientific and medical establishments. So there we have the government, clearly at fault, as we have said, but science and medicine, I agree with that. And one of the things they say is this monomaniacal focus on diagnostic testing using nucleic acid and antibody-based serology tests. And they say this just basically it shows an ignorance of viral host dynamics. And of course, we don't know for the most part what the dynamics are. The assays are not the problem, but we just don't understand the dynamics. And they say
the medical training thing
that's
interested me
that's very
because
interesting.
If I correct me if I've got this wrong, but what they're basically saying is that medical training has evolved in a fashion that trains individuals to, based on presentation, shunt people into a certain pathway of treatment.
That's right.
Rather than training physicians to think on their feet independently. Okay. It reminds me of analyses of why the U.S. was so successful. U.S. troops were so successful in the Second World War relative to other military forces that were so rigid in command structure that individuals had to follow the command structure and couldn't think on their feet. And this is actually comparing the German military with the U.S. military. The U.S. military was successful because the GIs were very innovative, okay, and had permission to implement innovative solutions to things. And what these guys are saying is that sort of creativity is being discouraged in the current medical training. Right. Did I get that right?
Yeah. They say we need to embrace old-fashioned, basic science-based medicine. Don't just shunt the patients into these clinical pathways. They say basically physicians are becoming intellectually unmoored. Young physicians, uncomfortable without a guiding pathway document in front of them, now feel intellectually unmoored because they were trained to have this pathway, and now this virus, we don't have a pathway, and so And in fact, they close by saying in 1910, Abraham Flexner decried the sorrowful state of medical education and academic medical practice in the U.S. and its divorce from fundamental scientific principles. 1910, this led to a complete revision of medical education and training based on direct application of scientific research. 100 years later, COVID-19 has deepened his call for reform. I remember that. Well, I don't remember it, but I remember it being talked about.
I suppose the good news is that in these areas, as well as several others, the pandemic is exposing flaws in our societies. And it's up to us now to recognize those flaws and address them.
Right. In the same way that, consumer product failures bring about changes in how things are made and done, this does that. And Rich, what you were saying about the ability to be flexible and creative also, to me, points out the difference between some universities and maybe others, like University of Illinois, where everybody was put to the task and one group developed the app and one group developed the assays and, all that sort of thing. And another thing that resonated with me is many universities and medical schools have actively discouraged research on the live virus, citing public health safety concerns and the lack of appropriate biosafety facilities. And there has been no wholehearted effort to rapidly build such laboratories or to pursue a moonshot project. So we have a BSL-3 where you can do the assays and things with cell culture, but our ABSL-3, our animal BSL-3, where you could do the mouse studies or other studies, was closed down some years ago because of nobody was using it, and it's really expensive to maintain that. And yet there's no interest, despite lots of pleas, to bring that back online so that some of these animal studies could be done.
That's terrible. Yeah. That's terrible. So I have to say that in January, Ian Lipkin wanted to bring the virus in, and he was told he couldn't because it's too dangerous. I said it was fine, and other virologists, but they found people who said it was too dangerous, probably lawyers. And I had to argue really hard, and actually the dean of the medical school and I had to have a conversation. The provost had to have a conversation. I had to emphasize how important this was. So this is absolutely correct.
This is you as being on the biosafety committee, right?
Yeah. I mean, we reviewed it and we said it's absolutely fine. And then some other committee that they convened said it's not. And there are a lot of lawyers on that committee.
I think that they talk about exactly the same part that Kathy's talking about with, the lack of laboratories and things like that. And I could imagine someone looking at sort of that paragraph and the previous and saying, well, we haven't had time to figure these things out yet. And so I hope there are two messages that people get from this. One, the importance of basic science, because we can't figure some of these things out without having a background in basic science. And with some of these things, we haven't figured them out yet because we may have realized, oh, there was a basic science problem we never knew the answer to. And I hope that we actually look at these things as okay, so how are we going to have a plan to reopen the ABSL3 in a pandemic and really try to learn from these things.
Right. I mean, and just think about it. China built a hospital in a week, right?
Yep. Yep. All right. One more thing from this article that really resonates. All right. They say people are dying not of infection, but pneumonia, renal failure, thrombosis that reflects the body's innate immune signaling. Okay. Of the $3.6 billion that NIH has reprioritized for COVID-19 related research, less than 3% of this money has gone. 40% has gone to diagnosis. Less than 3% has gone to the National Heart, Lung, and Blood Institute, despite the fact that lungs, heart, blood, and kidneys are the very organs responsible for death. I mean, I don't know. This $3.6 billion, I'm not seeing any of it. That's a lot of money, and I'm not seeing any of it. I'm told that we have to wait for our money now, and why? So there's a big problem, and so I'd love to get.
That's a really good point. Is that what kills people is not the primary virus infection.
That's right.
But the systemic response to it, and that's where a lot of focus should go. That's a really great point.
All right,
yes, innate immunity. Yes, definitely.
All right. Yes, Brianne.
We made Brianne happy.
I mean, it's just well, as you would say in Hebrew, fakakta. One of the Hebrew words I have learned from my colleagues. All right, let's do a couple of emails here. I will take the first because it's about my colleague at Columbia. Alok writes, "Hi, please comment on this article by Dr. Siddhartha Mukherjee." It's a New Yorker article entitled, "How Does the Coronavirus Behave Inside a Patient?" And Mukherjee, of course, the author of The Emperor of All Maladies, all about cancer. Really great book, Pulitzer Prize winner, love it. And so Alok gives us a few sentences. Quote, 3 questions deserve particular attention because their answers could change the way we isolate, treat, and manage patients. First, what can we learn about the dose response curve for the initial infection? That is, can we quantify the increase in the risk as people are exposed to higher doses? Second, is there a relationship between the initial dose and the severity of disease? That is, does more exposure result in graver illness? And third, are there quantitative measures of how the virus behaves in infected patients? For example, the peak of your body's viral load, the patterns of its rise or fall that predict the severity of their illness and how infectious there are to be others. All the answers to these questions to be decided. All right, so I was waiting for him to say that dose influenced disease, and I'm so glad he did not, right? Because this is something that I would expect that he might do. I think these are important questions and really relate to the opinion article that we talked about. We don't know a lot of fundamental stuff about how the dynamics of the infection. Very few people are working on that. They're mainly working on diagnostics and other things. So I agree that we don't know the answers to this. I'm not sure how this would change the way we isolate, treat, and manage patients, though. So I do think we need to know it, but I'm not sure how important it is.
Certainly the concept that I think falls within the purview of his third question here, the concept of when are you contagious?
Yeah.
Relative to, your symptomology or the virus load. That's something that we've talked about a lot. And the more quantitative, the more experimental data we have on that subject, the better. And also, in fact, this dose response curve that, how much virus does it take to infect you? I think is an important
That's
question.
important.
As well.
I agree. But
that's important. There are some data out there from older studies with There was a study that was pointed out to me from the original SARS that came out with a number of on the order of 250 infectious units being on average generally an infectious dose. But the point is well taken. There's not a lot of data out there, and these are things that would be useful numbers to know.
P.S. Another sentence from the article, quote, as a graduate student, I was trained in viral immunology, unquote, end quote. Sorry. Thank you.
That's usually what I say.
All right, Rich, can you take the next one?
Charles writes, hello, TWiVers. Great job on the Pants on Fire fact check. And he's referring, I believe, to the piece we did last week about the Yan article claiming that the virus was created in a lab. Not many people give as much of their time to the public good as you do. That by itself is enough to make each of you very special. TWiV is special in that as a group, it's as good as the sum of its parts. That's high praise. Most groups are only as good as their average part. Some, like the current GOP, are only as good as their worst part. And links to a PolitiFact, I love PolitiFact, links to a PolitiFact article that fact checks the Yan article and comes to the same general conclusion. Oh, actually, they give it their worst rating, which is Pants on Fire. Thanks, and please keep up the great work, Charles. We say in barbershop singing that a quartet is only as good as the worst singer in the quartet, which is really a pretty depressing situation.
Yeah, it's and, there are some other articles popping up that are similar that just repeat the same thing and with different viruses, with different bat coronas. It's just crazy. It's just crazy. By the way,
however, I do notice that it seems to me that in the long run, although these get a flurry of activity, in the long run, they don't get a whole lot of traction.
No.
There was remember Plandemic, the movie?
Yeah. Oh,
it was a big flash to start with.
Oh my gosh.
But never really gained much traction because there's a tremendous amount of pushback. And even though they get a little activity to start with, or even a lot of activity to start with, I'm pleased with the fact that in the long run, they don't get a lot of traction.
Yeah, they have their sort of ardent followers whose opinions are seemingly not going to change. But outside of those ardent followers, there doesn't seem to be a whole lot of uptake.
Yeah, as a matter of fact, the four of us discussed this document a week ago, right? And so I cut out that discussion and made a separate YouTube video of it. So it seems to be being viewed. So
it's such a blatant example of this sort of nonsense that not only has the phony science, but the sort of bad will behind it, with the connection to the Steve Bannon and the rest of them.
It's really sad.
Yep.
I mean, in fact,
I think.
Go ahead.
No, I was just going to say it would be good if you could put the link to that YouTube in the show notes. People could just point right to that if they need to.
Will do. So it turns out that our discussion was about 29 minutes of that, which I think is okay, right? That's not too long.
Mm-hmm. No, that's good.
Brienne, can you take the next one?
Sure. Bill writes that there's been wide rebuttal online to this Dr. Li Meng Yan conspiracy theory, and he also gives us the PolitiFact link, as well as a link to the AP, Newsweek, The Independent, and two links from the Tech Times. So it certainly seems like we are one of many doing this fact checking.
The more, the better, right?
Yep.
Absolutely.
Kathy, can you take the next one?
Suzanne writes, I believe there is a case to make against screening tests for mass gatherings. The false negatives could create spreading situations in places where people might crowd together and choose not to wear masks because of a false sense of security. The false positives could at the very least be a hassle. On the other hand, they're perfect for schools and businesses where people are generally in smaller groups and can be tested multiple times during the week. So I think this is true. However, if you're going to have mass gatherings and have no testing, that's even worse because you're going to have a lot of asymptomatic positives that are going to be there. And I'd bet that there's going to be a lot more asymptomatic positives than false negatives, or, people who would test false negative in a test. So I think, not having the mass gatherings, but if you must, this is just my opinion, a few false negatives may be less of a problem than the many untested asymptomatics.
Totally.
I agree with you, Kathy.
Totally.
To me, the key word in all of this is mitigate.
Yeah.
Okay. People tend to think of all this stuff in black and white terms. Okay. Yeah. It's either all droplet or it's all aerosol. Okay. You're either going to die at 6 feet or you're going to die at 10 feet. No, it's all a matter of probability. Okay. And the more you can do to decrease the probability, you can't make it zero, but you can decrease it, the better off you are.
Yeah. And I would hope that people would not sort of see that they have been tested and then use that as an excuse not to wear masks. I think that if you're in large groups of people, if you're in a lot of situations, you need to be wearing a mask.
I mean, the test, if it were done in this kind of situation, should come with a caveat saying, if you test negative, it could be a false result, and therefore you still need to wear a mask. You may be asymptomatic and a false negative, and if you were tested again tomorrow or the next day, you might turn up to be positive because your virus levels would be high or the test would be done differently. Yeah. So, I think but are people going to follow those kind of caveats? I don't know.
I just agree totally. It's just, if you don't test at all, then you're going to be much worse off than the few false negatives. But Michael thinks that he's only going to have 2% false negatives with his, and the FDA says they're okay with anything over 80%. So his is even much better. Jake writes, misinformation is still rampant. Hello, TWiVers. I work as a machinist, and as my particular job is rather boring, I listen to a number of podcasts. I was listening to The Indicator from NPR the other day, and they had Greg Ip on. He was discussing an article he had written for the Wall Street Journal, in which he claimed that the lockdowns were too blunt and we could and should do a more targeted approach. He made a number of claims with dubious legitimacy, but the one that made my head explode was his claim that there is no reason to keep schools closed because kids can't get sick. I just couldn't believe there are still people who think this is true, and there was zero pushback from the interviewer. This is exactly why we are in the mess that we are in. Anyways, keep up the good work. Every week I learn a little more and understand a little more. Jake. You're absolutely right, Jake. The data are clear. Kids get sick, maybe less frequently than older kids, but they get sick, they get infected, they transmit. I don't know why this is even an issue.
People misunderstand this. If you look at a curve of mortality versus age, you see very little mortality in, and for that matter, morbidity in younger age groups, more in older age groups. And people misinterpret that as thinking that means the people in the younger age groups aren't getting infected. Not true.
Right. I feel like we can remind people every day possible that infection and disease are not the same thing.
Right.
Just because you are infected with something doesn't mean that you're going to have symptoms. And I would, have people think about parents of small children who send their kids to daycare at the beginning. They often see both them and their children getting sick frequently as their kids are bringing lots of things home. So we know that it's not just about the kids transmitting among themselves at school. The kids come home to family members and other members of the community to whom they can transmit.
I also want to reemphasize something I've talked about before, and this notion that, kvetching about the notion that the lockdowns were past tense too blunt. Okay, and reemphasize that science is an evolving thing, and you can see it in this case. When this first hit, we didn't know how infectious the virus was. We didn't know how it was transmitted. We knew that there was a problem. And so under the circumstances, it made sense to shut everything down. Now we know a lot more about how it's transmitted. We know what appropriate procedures are to mitigate so that you can take appropriate targeted measures, which is being done. And to me, that's masks, distancing, and good hygiene. And by implementing those things, you can have a lot of activities that are commercial activities and social activities that are still ongoing and yet operate in a safe environment. And that's because the science has evolved and we understand it better.
Well, the lockdown was associated with a drop in cases in most countries. Yeah. And I want to point out that China, with the most severe lockdown, had less than 100,000 cases. That's remarkable for a country of that size. So don't tell me they were too severe. That's just expressing an opinion with nothing behind it, I think. Rich, can you take the next one?
Dawn writes, Dear TWiV, hello, I live in Wisconsin and have chosen to keep my second and third grade girls home for virtual learning this year as we continue to have increasing numbers of COVID-19 and because I observe that most in my community do not seem to hold to the reality of the pandemic as part of their, quote, belief system, unquote. I have a feeling that in Wisconsin, among other states, is in for a bad time in the coming months. My sanity is kept in check by my daily walks as my husband watches the girls and I listen to podcasts for some mental relief. You all are on the top of that list. My question comes from a conversation I had with another family member over the weekend. He, like my family, took the flu vaccine earlier than usual, about a week ago, since we have no idea where our state will be in another month or two. We know there is an effective window on the flu vaccine. My family member informed us that he planned to get a second vaccine in about 6 months just to make sure that he was protected. I said I hadn't heard of anyone being advised to do that, but this is not my area of expertise, and I wondered what the experts, you all, would say about that. Furthermore, he said he read an article that likened the immune system to a muscle that needs to be used to remain strong, and that getting an additional flu vaccine might bolster his immune system to be more prepared for a possible COVID-19 infection. I don't know the source of this article, but it goes against my understanding of the immune system. My background is in laboratory medicine and then dentistry. It's my understanding that, first of all, a person would not want a bolstered immune system or response, but instead a precise and accurate response to fight COVID-19, as it is the immune response that is really the thing killing patients in many forms throughout the body. Secondly, the idea of the immune system being compared to a muscle that needs to be used to stay strong does not ring true. I would appreciate if you could clear up these misconceptions that I believe some people may have about vaccines, how many flu shots they should receive in a flu season, and what, if anything, optimizes our immune system to fight precisely and accurately. I have always believed that it was a combination of good nutrition, good sleep, and managing stress levels. You're all great. I'm always impressed with how you handle yourselves and the integrity of the show. I look to you for the real deal when it comes to what science is telling us about both SARS-CoV-2, COVID-19, and vaccine progression. Keep up the good work. Warmly, Dawn in Wisconsin. Okay, so I'm going to do my 2 cents but my colleagues here, I think, have probably more like $1's worth on it. First of all, in terms of timing a flu vaccine, now is good. All right. I don't think now is too early or too late. And my understanding is that a second vaccine in the same season isn't going to do you any good. That's my understanding. But others can address this. And I think this notion that the immune system is a muscle is a bad analogy. Okay. And I think you've got it exactly right. Good nutrition, good sleep, low stress is where you want to be. And Dawn, you're using your brain just right. Go for it. Others?
I think I'll let Brianne take the muscle business, but I think remember the paper we did a while ago? The memory wanes in about a year for flu vaccines. So I think 6 months is probably not indicated. And I don't know any, although I don't know any advice to that extent, but it is true that older people who get immunized in August, by February, they can have waned immunity such that they get reinfected or infected. So maybe for the elderly, it might not be a bad idea. But they do have the higher dose flu vaccine now. Maybe that compensates for that.
Right. I was just going to say pretty much the same thing, that maybe for the most vulnerable, another immunization might be in order, but you could consult your physician on that. In addition to good nutrition, good sleep, and managing stress levels, I would say get vaccinated to the things that it's appropriate for you to be vaccinated against.
Absolutely. The muscle thing. So there is something known as the hygiene hypothesis, where there's this idea that your immune system sort of goes awry if you are not sort of infected with a few certain things as a child, that if you don't have certain pathogens in your life, then your immune system goes haywire. There's some interesting evidence there, and it really has to do with allergy and autoimmune disease. But I think people have sort of taken it to a different level in terms of how much practice your immune system needs, or how much experience your immune system needs. If someone is particularly worried that their immune system is not getting enough practice or is not getting enough use, I will tell them, perhaps sadly for them, that they are never going to be in a sterile environment. Their immune system is never going to be in an environment where it has absolutely nothing to do. So you really don't need to do anything extra in order to give your immune system some challenges. There isn't any great evidence that your immune system does need to be used frequently to remain strong. And I would agree that nutrition and sleep and managing stress levels are pretty key here. But even if you were really worried and wanted to make sure your immune system had something to do, fear not. There are likely microbes around that your immune system is taking care of. You don't need to give it extra. Is usually the way that I think about that. It is theoretically possible that you could luck out and get your flu vaccine with perfect timing, so that you had a non-specific immune response right when you got infected with SARS-CoV-2, but that would have to be sort of getting struck by lightning kind of luck. I don't think that's something that is likely going to be something that's going to happen to most people. And otherwise, I would just think about keeping your general health in good shape in order to keep your immune system in good shape.
I think that's
in
great.
terms of the exercise that your immune system gets on a normal basis, I'm reminded of something that I think about all the time, which is that when you die, you rot quickly. All right. And if you think about it, that phenomenon is what your immune system is fighting off all the time. Okay. So it's a constant struggle to keep you from rotting. And so you're getting lots of exercise.
Yeah.
Right.
As I tell my students, if ever their parents, are on their case and saying they're not doing something, there are lots of immunologic processes that they can tell their parents they're doing at any particular time. They're quite busy immunologically.
It's good.
Brienne, can you take the next one?
Sure. Ted writes, Alabama haiku. And he spells it HIGHCOO, which I assumed was intentional there. What's the difference? Ignorance and apathy? I don't know. Don't care.
That's perfect. I like it.
Fantastic. Thanks, Ted.
All right. Last one for today, Kathy.
Lisa writes, Hi, TWiV rock stars. Writing from sunny Sarasota, Florida, where it has finally cooled off to below 90 degrees. I'm feeling quite grumpy today as I send off the letter below to our local school superintendent, governor, and both the administrator and epidemiologist at my county health department. To me, the current policy referenced in the letter seems positively nonsensical. I'm interested in your thoughts. Keep up the great work. And dear so-and-so, I'm both a mom to a Sarasota County school student as well as a pediatric nurse practitioner in Sarasota. Since becoming aware of the seriousness of the COVID-19 pandemic, I've been working hard in both these roles to provide needed healthcare and to minimize spread. I believe all of us have felt some trepidation as the decision to allow in-person schooling to resume in Florida was made. I believe we all feel the challenge of weighing the educational and emotional costs of having our children not attend in-person school with the costs of potential increased cases and deaths by allowing them to gather together. Traditionally, once school resumes in the fall, we begin seeing an increase in illnesses. Despite all the measures with masks and distancing and hand washing, this has proven to be the case this year as well. My office is providing rapid SARS-CoV-2 testing, as current evidence suggests that children in particular may have varied and less serious symptoms with this infection, and that folks may be contagious either without or before showing symptoms. Our criteria to test is low. Fortunately, our results have been mostly negative, which is a reflection of the current lowered prevalence in this area. However, I believe that frequent testing with rapid results remains one of the cornerstones of our efforts to keep rates low. If we are able to quickly identify those who are infected, we can quarantine and contact trace to minimize spread. When we test patients in our office and obtain a negative result, we provide a return to school note advising of the negative test. Clearly, with a known exposure, current policy calls for a 14-day quarantine, regardless of a negative test. Today, we test patients and provide this note of a negative result as it seems best medical and epidemiological practice, both to prevent spread and to let our local schools know how many students are being tested and the results of those tests, as they also work to prevent spread. Today, the policy of the Sarasota County Schools regarding the return to school with a negative test, along with no known exposure, came to my attention. I thought surely the person telling me this policy must be mistaken, and so I called the school board to confirm. If a student has been home ill and has either not sought medical care or has not been tested, a note from a parent or healthcare provider is sufficient to return to school once symptoms are mostly improved and they have had no fever without fever reducers for the past 24 hours. However, if a student has been tested, even if they have had no known exposure and their test is negative, they cannot return to school without clearance and a letter from the Department of Health. This does not make sense to me. Asking parents who have sought medical care and testing to then have to jump through this additional hoop to get their child safely back in school seems to encourage exactly the opposite of what we ought to be encouraging. It encourages not seeking testing and merely sending their student back as soon as they are feeling better without knowing if they are infected and thus potentially still contagious. Regards. And then she adds, hello again. As an update to the email I sent yesterday to the superintendent of our school system, the epidemiologist, and the administrator of our county health department and our governor, I received a reply from the epidemiologist at the health department. They are changing the return to school policy so that students without a known exposure are able to return to school once they're afebrile and asymptomatic for 24 hours, and they will not continue to have to obtain a clearance letter from the health department simply because they were also tested for SARS-CoV-2. Obviously, this is for students who test negative. Those with either a known exposure or a positive test will be subject to either a 10 or 14-day isolation or quarantine, regardless of test results. It felt good to be able to use science and logic to affect a policy change at the county level, Lisa. So
Yay.
Nice.
kudos,
Yay.
congratulations.
Yeah.
Yeah.
Very good. Great job. It's sometimes you have to just try, and it may work.
Right.
You won't know unless you try, though. Great job. I really like that. You may notice that my far-UVC light went off in the background. That's a little LED that only has a 2-hour capacity. To do something about that. Kathy, you seem to have a pick of the week today.
Yeah, I had picked one and then another one came up that I just had to share. So this is just for fun. I think everybody needs a little bit of fun. So there's these Comedy Wildlife Photo Awards, and you can click on them. They were in The Guardian. And then when you get to that page, you can click on the eye and it will bring up the captions for each photo. And then there's forward arrows to go through. And if you're not smiling by the end of this, you need to start over and look at them again. But they're just really entertaining, I think.
Great.
Then the second link is because I have the astronomy picture of the day as my home screen on my browser. As soon as I went to browse for something, I saw that today's is the International Space Station transiting Mars. And it's the fascinating story down below it about this person setting up. Yeah, it was only going to be visible along a narrow stretch of land, 90 meters wide. The ground velocity was going to be 7.4 kilometers per second. And this person used a standard camera, a small telescope, an exact location, an exact direction, sub-millisecond timing, and created a video. And he got this image of the space station in front of Mars. It's just fabulous. So
Very cool.
Check it out.
Cool.
That's awesome.
Neat. That's amazing.
Both of these
photo.
are terrific, Kathy.
Yeah.
You guys want to start picks again?
Sure.
Yeah,
Let's
sounds
start.
good.
Let's start Friday. I'll add it in and we'll do it. I think people can use some picks. I
think I've already got one in here for when Dixon is
That's
around.
right. Friday should be
Kathy, Kathy gave me.
All right. So that is TWiV 666. So Kathy, what's special about 666?
Well, I looked it up, and of course, there's what Alan had pointed out a while back, that it's a number of the beast. It's referred to in the book of Revelation. So you can check out Wikipedia. There's an Iron Maiden album and title track to that. But there's this sciencealert.com link that I put in for us that describes something about the Greek and the real meaning behind it. But if you want to avoid all of that, 666 is the sum of the first 36 natural numbers. So it's a cool mathematical number as well.
That's neat. Very nice. All right. TWiV 666. You can find the show notes, letters, links, microbe.tv/TWiV. Send your questions and comments to twiv@microbe.tv. And if you would like to support us, microbe.tv/contribute. There are a bunch of ways there. There's PayPal, Patreon. There's an Amazon affiliate link, which means you click on it and then you buy your stuff at Amazon. We get a percentage. It doesn't cost you anything more.
And if you start to buy things at Amazon and then you remember that there's this thing, you can click on it and your cart will transfer and you can just keep going.
So
at least I think that's what happens.
It's good for everybody because we get a little money and you don't pay anything extra. So it's a good cause. And then there's Cafe Press where you can buy stuff, swag, right? T-shirts, mugs, everything, which people seem to like very much. And most of that goes to Cafe Press. And that's really so you can show your love for TWiV. We get just a little bit, $1 from each purchase, but it's still cool. All right, so microbe.tv/contribute for that. Rich Condit is an emeritus professor at the University of Florida, Gainesville. He's currently living in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. And I learned something new today about UV light is not purple.
Yeah, I did too. Brianne Barker's at Drew University on Twitter, BioProf Barker. Thank you, Brianne.
Thanks. It was great to be here. I learned a lot too.
And I am Vincent Raccaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Raccaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.